Hey guys, we're going to be talking about polymerase chain assembly, also known as PCA. This is a way that you can synthesize genes, small or large, from a bunch of different oligos. It can be pretty expensive for small academic labs, but it's routinely used in industry. I've used it a couple times on smaller parts, and it works pretty well. You do have to be careful because it can be finicky about weird sequences. If you have long runs of A's and T's, like you might find in a terminator, it gets finicky. Or sometimes if you're working with inverted repeats, like a FERT site or a LOX site, that can run into troubles as well. Basically, it's a bunch of wobbles in parallel. So I'm just going to go ahead and paste in some wobble-like reaction. So this, you can imagine, would give us a wobble-like reaction. But we're actually going to have a couple of these in a row. So we'll just start off with these two reactions. So you can imagine that if we filled these out, this would extend in this direction, and that would extend in that direction, and vice versa. So now we get two little fragments of DNA, which I've filled out in a wobble-like fashion. But there's actually a little trick here, which is if this fragment also has a little bit of homology to our first fragment right there, then, as we may remember, we are going to be able to do a sewing-like reaction. So if you haven't seen the sewing video, go check it out. So let's just draw on a 5' and a 3' and a 5 and a 3, and again 5, 3, 5, 3. So what's going to happen is once we've done this first initial extension, we're going to heat up our reaction one more time, and we can start to pay attention just to the important oligos, which in this case are going to be the inside, or not the important oligos, but the important strands, which in this case are going to be these inside strands denoted by our X's here. So we're going to ignore the top and the bottom strand. I should change colors, it's a little confusing. We're going to ignore these strands, that strand and that strand. And we're just going to look at these inside strands. So let's pretend that we're now heating this reaction up. So we're adding heat. We're going up to 95 degrees. Everything denatures, and now we have a bunch of single-stranded DNA. So we're going to put this single-stranded piece of DNA here. We have our 5'. And this single-stranded piece of DNA is going to be below. And now we'll just add in a few colors and make everything make sense. So I'm trying to line this up so we have our overlap region. This is 5', 3', 5'. That's a 5. I apologize. Oh wait, oops. I'm getting ahead of myself here. That is not a 5. That is a 3. So we have a 3' and a 5'. And if we wanted to break it down even further, this little purple chunk is the product of our original wobble. So we had our original homology region there, and then this little strand of DNA is the product of a wobble too. So we have a homology region right there, so we can track that up to there, and the same here. But now we're ignoring these. That reaction is in the past. That's already happened. We're going to be looking at this overlap region. And again, just like a sewing PCR, we have a 5' to 3'. We have an overlap, and that 3' can extend that overlap, and it's going to polymerize in that direction. Same thing is going to happen down here. We have an overlap. That 3' is going to extend that overlap in that direction. So that's how we get our first little bit of DNA. Now, since we put a bunch of oligos in, a bunch of primers, we already have extra DNA to amplify this reaction. So we extended this chunk down here, and now this primer right here can come down and start to amplify it even more. So if we had a primer there, we're going to amplify in that direction, and the same thing is going to happen with this primer. And we really start to amplify this part. So it's a really great technique for synthesizing stuff. And I'm going to really quickly try to go through it one more time. I'm just going to do the Cliff Notes versions, though my Cliff Notes always end up being a little bit more detailed than I mean them to be. So I'll try my best to make it simple. So just going to scroll down a little bit more. So we have a piece of DNA that we want to synthesize. We have the sequence, but we don't have the physical sample. So the way I would do is I would break it down into 3 different homology areas. I guess I'm going to be talking about how to design your oligos. And then all you really need to do, so once you have your strand of DNA, you have your 5', 3', 5', 3'. So all you need to do is design your oligos for this PCA like you would a series of wobbles. So this oligo is going to go to there, and then we're going to have the reverse oligo with that homology region, and then it's going to continue going all the way over to that homology region. And then we're going to be able to do the same thing. We're going to have an oligo going from there all the way to there. And this pattern can continue. You can add more of these if you want. And so this is how you would design your oligos. You have these homology regions right here. So homology, homology, and homology. And then you have these kind of gaps between the primers. Your homology should be about 20 base pairs. Of course, if you have a low GC content, you're probably going to want to bump it up. But 20 base pairs usually works. And let's just kind of see what happens when we run this PCR. Well, I guess we've kind of already gone through what happens, but we'll just do the Cliff Notes version. So we'll pay attention to these 2 oligos first. So these 2 are going to look like this. They're going to extend in that direction from the 5' to the 3', 5' to 3'. And then we can look at these 2. So these 2 oligos. So this oligo and that oligo have their homology region right there. So here's their homology region. Oh, I should have pointed out the homology region here. The homology region. And like our first time, these oligos are just going to extend. They're going to prime off of one another and fill out, giving us the products for a little sewing reaction. So mini sewing reaction right here. So if we were to look at the important ones, so again, we have the 5' and the 3', and then we have the let's see, the 3' and the 5'. In this case, these 2 are going to be the important strands. So we're just going to pay attention to those. And we're going to heat up our reaction. Everything is going to denature. We get our strand coming down. And we get our other strand right here. Slowly learning how to draw arrows with the thing. So 5 prime, 3 prime, oops, this is 3, and 5. And now this just fills right out in that direction. And we get a complete assembly. So sometimes what happens is this will run and you get kind of an inefficient reaction. You can tell by running it on the gel. And in that case, all you need to do is just add a few more oligos, just purify your reaction, add more of the outside oligos, and you can amplify whatever the product that you want to get is. So that is PCA. Very similar to wobble. It's like a bunch of simultaneous wobble reactions. And if you have any questions, let me know.